All right, hello everyone. Welcome. So just a quick warning, this was originally intended to be a little longer, so we had to cut it down. Our strategies, we're going to move through it very quickly, and any parts we skipped over too quickly, please ask us questions at the end, God willing. All right, so let's start. This is me. This is what I've been up to. This is my partner Savino here. This is what he's been up to. And this is the main idea we're going to go over. There are 958 unique flower samples of cannabis that we analyzed for 20 terpenes with chemical analysis GCMS. The main idea is we took these 20 variables times about 1,000 strains and used computer-assisted algorithms to reduce it down to 6 variables. From there, we used those 6 variables for 2 things. One, we mapped it to possible enzyme pathways that explain the theory of biosynthesis of terpenes in cannabis. 2, we used them to create the DiscOmic labeling system of which this presentation is named after. And additionally, hope for the future is that this reduced 6 variable group that can describe the terpene profile can assist medical doctors in recommending or maybe even one day prescribing cannabis strains to patients based on their disorder. So why are we focusing on terpenes? Just a real quick overview. We've got some data up on the wall here, and this is a cannabinoid profile. So each little box of the 4 boxes is a different strain, and you see replicate analysis from different cuts of that strain. CBD shows up in blue. Other than that, all you're really going to see is THC, green. We look at the next 4, we see pink. That's CBG, the precursor. That bud was plucked early, but otherwise, same story. We're just seeing THC. Last 3, same thing, THC. When we look at the global average in our 958 sample data set, 18% THC, but even though some strains are like half CBD, the average comes to less than 1%. So let's focus on terpenes instead. We see instead with terpenes, huge diversity. Not only do the strains agree with each other, meaning when you test the same strain twice, you get the same or a very similar profile. You also see diversity from one strain to another. It's very hard to confuse these profiles with each other. You can see the replicate tests show distinct replicate profiles. This one up here, Juicy Fruit's a great example of how when you test the same strain, you're going to get very similar profile ratios. Honestly, this goes on forever. And also what this opens the door for is fingerprinting. Terpene profiles are really unique between strains. There's a lot of strain fraud in dispensaries. If Blue Dream's selling great, I'll grow whatever I have and call it Blue Dream, and how's the bud tender going to know? With a terpene-based system, you can see a match and based on actual chemical science and say this was actually, the same strain. So the methodology's got 3 phases. Phase 1 is chemistry, chemical analysis, how do we know what terpenes are in what. Phase 2 is computer-assisted algorithms and data analytics, and phase 3 is putting it all together, making the enzyme pathways work. So phase 1, or stage 1 of methodology is just chemical analysis. So for those of you familiar with chemical analysis, it's a chromatography, GCMS, headspace sampler, so it's a gas phase sampler, and we're just basically evaporating the terpenes off the cannabis sample and separating them and quantifying them with a mass spec. We had to really reduce the data because there's a lot of not so defensible data out there in the cannabis market, and that would have thrown off our phase 2, our computer-assisted algorithms. So we had to really limit it to just a couple labs that we had to audit very closely. Total data set was 958 at the bottom of the screen here. If you're familiar with terpenes and cannabis, I'm sure you're familiar with all of these 20 at the bottom. And so a little bit about our methodology here. We have 4 techniques: LC, UV, LCMS, GC liquid injection, and GC headspace. They all have advantages and disadvantages. We chose the one all the way at the end, the GC headspace. Main reasons being it's a very clean loading with low interference, so you get good, accurate data. GCMS liquid injection, which I know some other researchers are very fond of, has some maintenance and analyte breakdown issues, but otherwise is very comparable. The LC I would honestly stay away from. And this is what a chromatogram looks like for those of you familiar with chemical analysis. We can see the numbers here, 1 through 36, are the different terpenes that were isolated from a cannabis sample. And I'll pass it to my partner for stage 2.
So there's been an exponential increase in the amount of data associated with cannabis chemical profiles and genetic profiles. And as such, a lot of traditional, as we've seen today, a lot of traditional chemometric and metabolomic methods have been applied, including PCA, LDA, linear regression. And these are great, and what we want to do is take these and put them into a form that could be better understood and absorbed by the general public. So some previous studies have found that strain name, there's no significant terpene profile clustering around strain name, so strain name is not a very good predictor of what the final terpene profile is going to be. And as well, last year we found that the strains can be subdivided into overlapping groups, as we've also seen here, based on dominant terpenes. But what we wanted to do is, instead of basing it on the dominant terpene, we base it on the ratio of some of the several dominant terpenes to each other. So we started with self-organizing maps, which use basically a bunch of iterations of principal component analysis to group the samples into a higher dimensional space. use this to see which ones appear to be related to each other, and test it out with some k-means clustering. Use that to translate into our label. And just to give you a quick overview, this is a self-organizing map. Red indicates high concentration, that light whitish blue indicates low concentration. As you could see, high CBDA strains and high THCA strains are on opposite sides because they occur differently in different places in the data. They're organized by their chemical profile, and the profiles are in separate places. There's a little bit of crossover in the middle, but for the most part, they exist on different sides. They're different genes that code for each one.
All right, and so now we're on to the third stage of the methodology. Once we have the chemical data, once we play with it, or let the computers play with it, we got to map this together and explain how these terpenes are being made by enzymes. So step one is we needed peer review literature on enzymes, terpene synthases in higher plants, just to get us in the ballpark. And so this is just a screenshot from a database we've built. As you can see, there's these columns in the middle, output one, output two. Some of them just make one terpene, some of them make a family, some of them make them in fixed ratios, some don't. So that's part of the complexity of mapping this out. All right, and so the idea is to filter through all of those entries and match them to the data that we're getting from the computer-assisted algorithms and generate a map that would look something like this. This is more hypothetical, but a more defensible map that we can link directly back to the data. So, now that we've overviewed the methodology, let's actually go through the data. Stage one was chemical analysis. So we have 958 samples times 20 terpenes, which is 20,000 data points. We started with a standard approach, statistics, right? We do averages, standard deviations, global maxima, and see what it looks like. We did get 11 terpenes that are at significantly higher levels than the others. And I listed them here at the bottom, but it's more fun to look. The blue is the mean. Blue plus red is mean plus standard deviation. Blue plus red plus yellow, we threw the global maximum on top of that. And so when you tally them up, you can see that statistically there's 11 players that are going to kind of tower over the others in these profiles. So you think, okay, we got our 11 that we're going to look at. There's some issues with that. Focusing on high concentration terpenes is a problematic approach. Terpenes are not known to have a same medical effect on the human body. Just because a milligram of linalool does something doesn't mean that the active dose for another terpene couldn't be much higher or much lower. So looking at the absolute concentration doesn't really give us a good idea of why things are medically efficacious. Additionally, there's been a lot of reports that terpenes have very complex metabolic pathways and very complex medical effects on the body, and that small concentrations of other terpenes can affect the way our body's processing the higher concentration terpenes, what some people call the entourage effect or synergistic effects. This means we need we need to go past statistical analysis and do something a little fancier so we can look at the complexity of this 20 variable data set. That's where you come in.
All right, so we turn to the self-organizing maps. Here we see the results of the terpenes run in the self-organizing maps. As you can see, the alpha-pinene, beta-pinene occur most often together, as well caryophyllene, humulene, and nerolidol. And over here we see limonene and linalool occurring in a very tight correlation together, and the terpenes occurring together. And as you can see, they don't occur in the same space. Myrcene and ocimene occurred in different spaces, but there is some overlap, and we'll go over that in a moment. And this is just the normalized data plotted plane, and all we see is some of the points seem to cluster around axes. And here we've run it into k-means clustering. And as you can see, this is the limonene linalool group versus the terpenes group. And there's 3 major clusters that separate very nicely, and this indicates 3 different genetic variants that are producing different relative amounts of these terpenes. And again, we see this when comparing the pinenes to the terpenes. Here we have the myrcene ocimene, so there's a group that produces myrcene alone, and there's a group that produces myrcene and ocimene together. And here the sesquiterpenes, caryophyllene, humulene occur in a very tight correlation with about a 1:3 ratio. The others, not so much. In order for this to work, there have to be valid methods. The methods have to match very tightly. There have to be proficiency rounds, and make sure that all the data can be aggregated. And also, we want a diverse data set, genetic, geographically, as well as environmental, growing conditions, all that stuff.
All right, so now that we've looked at the patterns we saw from the computer-assisted algorithm analysis, let's look at how we can make sense of this. So just a quick review. When you have a theory where there's 2 compounds coming from one enzyme with one precursor, we expect to see positive correlation, right? As that precursor 1 at the top, as that concentration increases, the concentration of the 2 compounds at the bottom is going to increase in a fixed ratio, so we see positive correlation. When we see two compounds that have a common precursor but have two enzymes, we will see a competitive situation where their sum is relatively constant. As the amount of the precursor is competed by the two substrates, we see a negative correlation in the concentration of those two compounds. So using these principles, we looked at Croteau's 1987 paper to kind of create a landscape, if you will. How do other plants that are more well-studied than what we're looking at, cannabis, make their terpenes? And we tried to map out a cannabis map from the materials in the Croteau paper, and we got something that looked like this. Now you can see the colors don't seem to correlate in these competitive groups like they should have a moment ago. And so this is, the genetic variation from one plant to the next, causing some difficulty. So if we zero in on the different axes that my partner was talking about before he was talking about ocimene, myrcene. Myrcene can appear alone, ocimene can't. There's two real groups, one with both and one with only one. Best way to explain this is two enzymes, one that produces just myrcene and one that produces myrcene and ocimene in a fixed ratio. Assuming that they are of the same gene and that they are shuffled alleles, we're going to see, homozygous and heterozygous plants of different combinations of this, giving us three ratios, which is what we saw: myrcene only, one ratio of myrcene ocimene, which was lighter, and then a heavier ocimene favoring ratio. Let's look at the next one. Unfortunately, they get much more complicated than that when you look at terpene families. So the terpenine family had four. We looked at three terpenes, alpha, delta, and gamma, and then we looked at carine. And they seem to have the common precursor that moves their concentration together, which here we labeled as 2, but we're hypothesizing to be a terpenyl pyrophosphate, or some sort of terpenyl cation. On the other side, from the common precursor, we see that an enzyme moves it the other way towards precursor 3, which we're hypothesizing is linalool pyrophosphate, because it's known to be a precursor to both limonene and linalool, which co-occur in our data set very tightly. And again, using literature in other higher plants other than cannabis, we're hypothesizing that the common precursor they're competing over is geranyl pyrophosphate. Now, as for the last group, orange versus green, it's more difficult because the pinene don't show a very clear correlation with any other group. We at first thought this was anomalous, but when we started looking through that literature database, we saw that pinene is synthesized a very large number of ways. There's a countless number of ways to get to that product. And in other plants, there's a great diversity of those enzymes that can get you to that same product, so it's not easy to draw these linear correlations like we were doing earlier with a positive and negative correlation. Conversely, on the orange side, we see enzyme 4 creating caryophyllene and humulene, which we hypothesize uses the precursor farnesyl pyrophosphate. And we think this agrees with our data set, showing a positive correlation in a fixed 3-to-1 ratio between caryophyllene and humulene across the data set.
So all this comes to the labeling system. Our goal was to create a label that would be simple and intuitive for the average consumer. It would provide a standard approach to product categorization and selection. It would take something that has been qualitative, the choosing of strains, usually by smell and taste, and turn it into something more quantitative and non-subjective, like we would see with a nutrition label or with a chemical hazard label, something easy to recognize, but also quantitatively based. So here's the label, and here's sort of how it's set up. So we have the thickness of each radius in a ring. That corresponds to the normalized concentration of that subgroup, the groups that we talked about before, which are splayed out here. And so the rings can be either solid or split, depending on which of the clusters it falls into. And if they are split, then the ratio of each color corresponds to the ratio of those two groups with each other. And what does this translate into? So when we run the actual numbers through this system, what we get are these color-coded labels that are very indicative of the terpene profile. And as you can see, you can match strains using this. You can see the Gorilla Glue has almost identical labels, whereas the Casey Jones and Cherry Diesel are very, very close to each other. And they are very similar. And if one were to run out of Casey Jones and want another strain that was very similar, and well, they could go for that Cherry Diesel. And these are sort of the major, 27 major groups, since each of the concentric rings has 3 possible formats. So here are the 27 possible. And then here are a histogram of how many samples in our database were in each of the groups. And as you can see, there are some groups that where most of the data falls, and there's some that weren't represented at all. And this could mean either these are areas where breeding needs to happen, or areas where it's impossible because of the underlying genetics.
So that concludes what's actually been done. Let's talk about where we want this to go.
3 minutes.
We have on the left here, you see the 958. That's the database we've been talking about this whole time. On the right is a small medical paper database of stuff not behind a paywall that studies either a cannabinoid or a terpene and its effect or efficacy on a known human disorder. And the idea is, with this labeling system, we're hoping to facilitate doctor recommendations and possibly prescriptions of specific cannabis strains with specific chemical content to specific disorders. Of course, that's the hardest part out of all this is not the chemistry, but the medicine. So this is just a screenshot of what this database looks like right now. We only have about 500 papers in there. Again, they can't be behind paywalls and they study cannabis, so, slim pickings. But, we see on the left all the publication data, and on the right you see those green check marks. They're tracking chemical compounds. So on the far right, you're seeing cannabinoids, THC, CBD, all that stuff. In the middle, we see which disorders being looked at, and then further to the left, we're looking at what terpenes the study might have looked at. Now, a lot of these studies didn't look at cannabis because, the history is not easy to do clinical trials on cannabis. So a lot of this is essential oils. A lot of this is other compounds that contain these terpenes. And so when we output these 501, we get a very slim pickings output chart. But the idea here is that you see the terpenes in the first column, and then the disorders across the top. The number represents the number of peer-reviewed papers from our database that indicate a positive effect for that compound on that disorder. And so right now we're in the double digits. We're nowhere. We need to go up orders of magnitude before this becomes an actual usable resource, or could even possibly be one. But the idea is to connect it to the DiscOmic system to make better recommendations. And so this is another form of output where we basically listed each peer-reviewed paper in a list, going down the left and right. And then to the right, we have columns basically populated when a terpene is one of the compounds of interest studied. And then we eliminated the cannabinoids for this purpose. And then we also programmed it to be color-coded to the dominant terpene group from our label system. So a resource like this, with many thousands or hundreds of thousands of papers, maybe, would be a much better resource for medical doctors to look quantitatively at what's actually in the strain and how that has statistically been proven to be effective against a variety of disorders in humans.
1 minute.
And that pretty much sums us up. Special thanks to DB Labs and MCR Labs and Leaf Therapeutics.
A round of applause. Thank you very much.